Hello everyone, Mr. Dunnan here, and today we're going to be looking at protein folding. Now, if you haven't caught my last two videos in particular, there was one on transcription, RNA synthesis, and translation protein synthesis. You really need to check those out first before you watch these. So there'll be a link up in the top corner. Click on that, watch those other videos, and then come to this one, and this one will make a lot more sense. The outcome of translation was to produce proteins, and those proteins had specific functions. So whether it's an enzyme or what have we got here? Insulin, antibodies, muscle fibers, and protein channels, they've all got a particular function. And we talked about in the last lesson, how do they get that? They have a specific structure. Now, how do they get that specific structure? Well, it all comes down to that sequence of amino acids that they have. That particular structure, that three-dimensional structure, really depends on all of these interactions between those amino acids. So whether it's ionic bonding or van der Waals forces, polar, non-polar interactions, all of those things will determine that overall 3D shape. So let's have a look at an amino acid in a little bit more detail. So here we have an amino acid, and it has this same structure no matter what amino acid we're looking at. So we've got 20 amino acids that we're looking at in our general biology course, in our eukaryotic cells, our organisms, and they all have this amine group, which is this one here, and a carboxyl functional group. So we've got these two groups, the amine group and this carboxyl group, and then the rest of the structure is the same, but what differs between amino acids is this R group. So I mentioned that there was 20 amino acids that the mRNA codon system codes for. There are 9 essential amino acids, so that means we have to include those in our diet, whereas the other 11 we can manufacture ourselves. So those 9 have to be in our diet. They all have the same structure, but what's different is this R group. So what's different about it? So here I've got this great image that shows the types of R groups that could be in those different amino acids. So for example, if we look at these non-polar R groups that also have a neutral charge, we have things like tryptophan, phenylalanine, glycine, alanine, valine, isoleucine, leucine, methionine, and proline. So they all have a similar R group in terms of their chemical properties, that they're neutral and non-polar. Then we have some amino acids that have a neutral but polar nature to them. And then we have some basic R groups and some acidic R groups. So because of all the different chemistry between these R groups, they can actually interact with each other and bond in a way or cause sections to fold if they're in a line. So if you're doing organic chemistry, you're probably going to learn a lot more about them than we will in this lesson. But for our, what we need to take away is that because of these differences in R groups, it will cause them to interact. Okay? So if we have a sequence of amino acids, that sequence might be different, which means those interactions are going to be different, and then we're going to get an overall different folding. But we'll go into a bit more of that in a second. We don't have to know which one's which. We don't have to know too much detail about the chemistry of the polar, non-polar, the acidic, basic, and all those types of things. We don't have to know any of that. All we need to know is that because of these things, we're going to get different folding depending on how they're ordered in the amino acid sequence. One last little thing that we'll get to before we go into some of the other sections of how we structure that folding is when these amino acids become close to each other during translation, we form a peptide bond. So what is a peptide bond? A peptide bond in this case is actually a condensation reaction where we have the hydrogen oxygen of one amino acid will react with the hydrogen of an adjacent amino acid, and then we release that water molecule, and that releasing of that water molecule covalently bonds those two amino acids together. They're now strongly bonded together. We call it a peptide bond. As we bond amino acids together in this way, we form a peptide. And if it's more than 30, we call it a polypeptide. And if polypeptides don't merge with any other polypeptides, we then eventually will get a protein. If they do merge with other polypeptides, we then just have a more complex protein. Here we're going to look at the organization of those proteins in more detail. So the first thing we look at is just the primary sequence. So the first structural element of a protein is just the sequence of amino acids that we have. And remember, they came from the sequence on the mRNA strand and the codons, and that came from the gene that we transcribed. So you can see a bit of a flow on here. Gene transcribed to mRNA, translated into a sequence of amino acids. That sequence can sometimes go through some post-translational modification, just like our mRNA can go through some post-transcriptional modification. We can add things like phosphorylation or glycosylation as well, which just changes that protein sequence just a little bit. And then some of those post-translational modifications are needed to make it fold in the way that we want. But the primary sequence or the primary structure is just that initial amino acid sequence. What's important to note is, to be specific, we actually say that a protein is made up of amino acid residues because they've lost that water molecule during that peptide bond. So let's have a look at the secondary structures here. Looking at the secondary structure now, this is actually the types of shapes that we get through hydrogen bonding of basically the sequence of amino acids. So it's not an interaction between the R groups, it's actually a hydrogen bonding between those other aspects of that amino acid. So if you think of that like peptide backbone, we then get some hydrogen bonding between those, and we get two main shapes. So we get hydrogen bonding causing this alpha helice shape, and we also get some hydrogen bonding which causes these beta pleated sheets. And I sort of draw them like this. So if you imagine our alpha helix is sort of this shape, okay, that would be our alpha helix. And then the beta pleated sheet would look like this. So the secondary structure is just areas of the amino acid sequence, the polypeptide, that forms these alpha helices or beta pleated sheets. That's the secondary structure. The tertiary structure refers to the three-dimensional structure of the polypeptide. So now we're looking at the overall folding of these alpha helices and beta pleated sheets within the overall three-dimensional protein. Now, this folding occurs from a number of reasons, but it's all mainly due to those specific R groups that we mentioned earlier. So for example, we might have a section of hydrophobic R groups, water hating, that all group together and form like a little section away from the solution. We might have water loving sections, so the polar sections that actually orientate themselves to the outside. We also have certain areas of different R groups that undergo disulfide bridging. We have some ionic interactions between acids and bases and form these salt bridges, for example. And these ionic bonding, some hydrogen bonding, some disulfide bridges, some van der Waals forces, all of these things start to interact together. These polar, non-polar, hydrophobic, hydrophilic regions that all start to interact together to form that overall three-dimensional shape. So if we go back a step, we remember all we have is just an amino acid sequence. It's the order of those sequences of amino acids with their specific R groups that then cause those alpha helices, beta pleated sheets, and then all of those interactions of the R group. And then when we have that, we have the three-dimensional structure. Once we have that, it can have a specific function. Okay, so having a look at this here, this is a website. You can see it on the screen. It's a really good way just to have a look at some of those interactions between R groups that I mentioned earlier. So if we just have a look at some here, so you can see these two amino acids cysteine, when they become close to each other, they form those disulfide bridges that I spoke about earlier. So if they're near each other, then you're going to get that disulfide bridging, and therefore they're going to bond together, and that's going to affect the overall shape of the polypeptide and therefore the protein. So you can see an example of that here. What else do you notice? You might notice this group of yellow amino acids, which are hydrophobic. So as I mentioned earlier, because of their hydrophobic nature of those R groups, they tend to sort of connect together to sort of try and reduce as much distance as they can away from water. So that's going to cause some folding in that region. And then you will notice that the hydrophilic regions then orientate themselves towards the outside of the protein. So we've got the hydrophobic R groups and their amino acids orientating inwards, and the hydrophilic regions orientating themselves outwards. These two molecules here, these two amino acids, we're getting ionic bonding, sometimes called salt bridges, basically between oppositely charged particles like lysine and aspartic acid. If we have more than one polypeptide, we call this their quaternary structure. And I didn't mention it in the previous slide, but the quaternary structure is just multiple polypeptides together. And so we could add those in here. And then here's an example of the hemoglobin protein. So we've actually got 4 polypeptides, which means we had 4 genes contributing to their sequence of amino acids. They fold in a certain way. They then join together in a certain 3-dimensional structure that gives it its function. So you hear me saying that a lot. Structure equals function. Thanks for watching, everyone. I hope that sort of clears up protein folding, gives you some information there. If you like the video, give it a thumbs up. YouTube likes it, helps me out. Make sure you subscribe. If you want more videos, you'll notice that they're coming out a little bit more regularly now. So next week, we'll probably just shift gears a little bit and look at DNA synthesis, DNA replication, as well as some factors that affect proteins as well. If you want more of these videos, check us out on Facebook. Make sure you like that page. You can also find us on anytimeeducation.com. Videos are really nicely organized on there, and there's some videos from my colleague as well. So check that out. And again, we should be seeing some other videos in the corner popping up. Watch those too. You'll really enjoy them. Bye.